Let's take a look inside the bacterial cell membrane.
Cyto means cell and plasm means colorless liquid fluid. Therefore, cytoplasm is colorless gel-like substance. This colorless, dense, gelatinous solution enclosed in plasma membrane. Cytoplasm is composed of water, which 70 to 80%, and also sugar, amino acid and salt. They all are important for, serve as a solvent for material used in all cell function. This compositions provides a milieu or environment for biochemical reaction.
Facilitates the organization of cellular structure.
And serve as a reservoir for cellular components and molecule necessary for cellular function. Bacterial cells lack membrane-bound organelles in their cytoplasm. The cytoplasm possess a range of specialized structure and molecules that enable them to carry out essential functions and adapt to various environmental conditions. Let's take a look one by one. Water provides medium for biochemical reaction and allowing the diffusion of molecule within the cell. Inorganic ion, that is salt, essential for cellular processes. Maintaining osmotic balance, this is regulation of water and solute concentration to maintain internal stability. Bacteria achieve this by controlling the movement of water across the cell membranes. By regulating enzymatic activity, bacteria utilize enzymes to catalyze, or given a speed, to wide range of biochemical reaction, essential for various cellular processes. Like metabolism, cell wall synthesis, DNA replication and nutrition uptake and utilization and so on. By employing diverse mechanisms, bacterial cells can efficiently regulate the movement of substances across their cell membrane, maintaining internal homeostasis and responding to changes in their environment. Ions play a crucial role in enzyme activity and membrane stability, and also contributing to osmotic balance and pH regulation. Each metabolite play a specific role in bacterial metabolism, help in generating energy for overall functioning, and physiology contributing to the overall function and survival of the organism. Depending on the specific bacterial species and environmental conditions, other components are also involved to play a crucial role in the cytoplasmic environment of bacterial cell. Inclusion bodies serve as a storage granules for storing energy or nutrients. Mechanisms of buoyancy regulation enable bacteria to optimize their position in aquatic environment. Bacterial ribosome is a cytoplasmic nucleoprotein particle whose main function is to serve as the site of mRNA translation for protein synthesis. The average diameter of ribosome is around 20 nanometer, constituting approximately 30% of the cell dry weight. Approximately 10,000 ribosome in a single bacterial cell. Collective presence of this ribosome contributing significantly to the granular appearance of the bacterial cell. Swedberg units indicates the rate of sedimentation during ultracentrifugation, depends on size, shape and weight of the molecules. S value is maximum for heavier molecule than the lighter one. Bacterial ribosome structure is 70S ribosome. The ribosome will be divided into two parts. The upper portion is large and the lower one is small. The large size of ribosome is 50S ribosome, where the small one is 30S ribosome. During protein synthesis, ribosomal mRNA bound to the small 30S ribosomal subunit, and a large 50S subunit of ribosome link amino acid by providing the polypeptide chain. Ribosome do not function on their own but it required as other factors. First is initiation factor. Translation of mRNA start at this point. At this initiation point the mRNA backbone is linked with nitrogenous base. Three collective nitrogenous pair are code for one amino acid, hence this known as codon which is represent amino acid. The ribosomal 30S subunit find the start codon on the mRNA strand. The initiator tRNA carrying methionine amino acid and bind to the start codon. Then the large ribosomal subunit 50S joins the small ribosomal subunit and forming a functional ribosome around the mRNA strand. The ribosome is now set to begin binding the protein during the elongation phase. There is the elongation phase during mRNA translation. In codon recognition, the ribosome attached to the mRNA at the start codon. As the ribosome reads each codon, a matching tRNA molecule, carrying the corresponding amino acid binds to the codon. New tRNA brings an amino acid to the ribosome and it gets linked to the growing chain. The ribosome moves to the next codon along the mRNA, shifting the tRNA and their attached amino acids from the A site to the P site. The last factor is termination factor. On this step, mRNA translation get terminate or stop. These termination factors ensure that protein synthesis stops at the appropriate place, and that the completed protein is released from the ribosome. Within the large ribosomal subunit, the A, P, and A sites play crucial roles in coordinating the binding and movement of tRNA molecules during the elongation phase of translation. Again, we look the process of mRNA translation initiation to termination. At the end, the last and most important component is bacterial genome. Bacterial genetic material typically consists of single circular chromosome, composed of double-stranded DNA. Part of this compaction comes through the formation of loop domain, where segments of DNA are folded into loops-like structure that are held in place by DNA-binding protein. Further compaction is accomplished through the process of supercoiling. The chromosome contains genome, which help in function to carry genetic information, including genes essential for its survival and reproduction. Some bacteria may also contain smaller circular piece of DNA called plasmid, which often carry genes that provide additional function abilities, like antibiotic resistance or the ability to metabolize certain substances. At the end, this is the summary of the topic.